Good morning, everyone. I'm John Castle, and I'm going to be talking about our increase in support for genomics and biomolecular sequence processing. Today, we're going to be talking about, starting with talking about biomolecular sequences. That's the area where there's been kind of the most happening recently and really the most progress to report. We're going to give you some motivation about why it's important to have support directly for biomolecular sequence processing. We're going to review some of the 12.2 features, including sequence representation, some common biomolecular operations, and the Wolfram language integration. And then we're going to be spending most of our time today looking at the new features in version 13, including support for additional bonds, for hybrid strands, for collections of sequences, and for visualizing all of these new features, as well as a variety of extensions, modifications, and other conveniences aimed to help you work with these larger structures. After that, we'll be turning briefly to the future of biomolecular sequence work, looking at annotations, lookup and alignment, and beyond. After that, we're going to turn to some our biomolecular entity support, or give a survey of some of the existing entity types, including for specific structures, such as genes and proteins, as well as entities that support configuration and really allow the other functionality to be richer by having access to data that allows it to reflect about its own type and extend its capabilities. And then we'll look to the future of those, discussing some updates and then potential biosequence bond type. We'll very briefly survey some related Wolfram function and data repository resources, just a couple of examples with some links to further resources, and then kind of turn back to discuss kind of this overall direction and how making our progress is really aimed to make move forward in a variety of facets. After that, we'll sum up and give some time for feedback and questions. So why BioSequence? Why are we working with molecular sequence, biomolecular sequences? And we noticed that kind of molecule had the ability to work with chemical entities and allow users to operate more on them more richly, but that this molecule representation extends beyond just notable chemicals to any reasonable molecule designated by the user. And so we wanted to provide a level of abstraction above molecules to conveniently allow common biomolecular operations, so that like sequence complements and but this would also allow us to manipulate the kind of the space of biomolecular entities that we had, so that when we could take our existing support for genes and then work with them as biological sequences, and yet also have a sense of integration, so that we could work with things at this abstract level, but still always have a clear tie to the kind of the molecular world. So in order to provide this, we introduced biomolecular sequences in 12.2. We offered kind of representation for DNA, RNA, and peptide sequences. And each of these had deep support for degenerate letter processing. So letters that could represent multiple physical sequences for when the data is kind of unknown experimentally. So here we see Y can be resolved to either C or T. And then we could sample kind of that space here. And we also wanted to have it so that BioSequence could be extended by users for the sequence types that they cared about. So here we're adding a Hajimochi DNA as an extended BioSequence type, and it has its own complement letter rules and its own alphabet. It borrows the DNA icon. And then by virtue of registering an entity store of an extended biosequence type, we can perform complements on Hajimochi DNA. We can access the Hajimochi DNA type sequence. We could do string processing on it and kind of all of the rest of the kind of the basic functionality that sequences have. So that release also provided all the common biomolecular operations, complements, transcription, translation, and back translation. And it also had some relatively deep Wolfram language integration. So in particular, we were thinking of that string processing was very important for biological sequences. And so that string operations could work directly on biosequences. And that there would be the string operation behavior would behave specifically for biosequences in some key ways. For example, if you're processing a biosequence, you'll get back a biosequence instead of a string. For functions that perform matching, degenerate letters would produce additional matches, so that here the W would match the T here across a variety of operations. And that if you wanted to have a specific match, you could say verbatim. So that verbatim would say, okay, give me this sequence, part of the sequence exactly, and allow degenerate matches in these remaining characters. And these same principles apply in functions that use rules, so that this match here will occur by virtue, again, of the W matching the T. Another key difference was that circular types would now allow wraparound matches, so that the C ending here and the A starting here would be registered as being contained. And sort of similarly here, the degenerate letter S covers both G's and C's, so that a sequence of S's followed by an A does match circularly for this sequence here. And similarly for string count and string replace. And now in circular replacement, the framework can alter where the start of the sequence is, as you know, that's not necessarily well defined. Now, circularity also means that positions that it takes wraparound positions. So we've taken the fourth through the first, or the A and the C, and likewise for dropping and for replacing. Similarly, wraparound positions, can be produced from operations that find positions. Now, beyond the string integration, a variety of other sequence operations are supported for biosequences, one would expect. So clearly sequence alignment is going to work in a way that it allows us to apply further operations in the way we would expect. Having given this kind of catching everybody up to where we've been, let's introduce kind of the new biomolecular sequence features in version 13. So the first is that additional bonds can be added between sequence terms. So here we're adding kind of this multi-hydrogen bond here, and we can see it joined together in this plot here. Now, bonds at the level of a sequence may represent a set of bonds at a molecular level. The multi-hydrogen bond in this A and T above actually has 2 hydrogen bonds connecting these base pair. Now, a number of such bonds can be added. So you can add these here. And to specify a number of bonds, it can be convenient to use this dot bracket notation as a shorthand. So here, this innermost parenthesis here matches this one, and so on. So that you're indicating kind of a level of nesting as you follow through the sequence of the structure. And you can support, describe fairly sophisticated structures this way using two different, kind of layers of kind of bond nesting at once. Another way to specify multiple bonds is to use a span for each index of the bond. So here we see the kind of a span from positions 2 through 6, and one down from 18 through 14. Spans of bonds that are evaluated to the simpler bonds that they're shorthand for. Now, by default, the bond that the user provided is not assumed to be of any particular type. If it's not provided, the bond type will be inferred for operations that require them where possible. So by default, additional bonds of DNA and RNA are of the multihydrogen type, which allows for both the standard base pairing and any other pairing that might typically be encountered. Now with peptide sequences, it matters what is being connected. The supporting peptide types, disulfide and lactam bridges, can only be connected between certain amino acids, and not all connections are supported. So here we see we're adding a bond, and by virtue of the characters here, it determines it's a disulfide bridge, while in this case determines a lactam bridge. While here we see it's unable to infer a type that's currently known. Now with these bonds, we can do some fairly nice things. So here are some example toxins that have some nice 3D molecular plots kind of given these bonds. So here we are adding kind of the bonds to this sequence here, and we see that it knows that they're disulfide bridges, and from there we can get a molecular mass and a 3D molecule plot here. And similarly for these other toxins here, can determine their bond type and get their mass and molecule accordingly. Now, we've also, in addition to allowing bonds, we've also introduced hybrid strands. Now, it's a single strand composed of many, motif sequences, possibly of different types, and they're bonded end to end with the primary linkage for those type of sequences. For example, DNA and RNA occur together in hybrid sequences, as they both have phosphodiester bonds forming their primary linkage structure, so that they can be linked, along their, their primary connectivity, kind of in an end-to-end way, forming a single strand. Here we see this strand together, and it's plotted here. Now, this is a shorthand that's happening here. So this is actually getting expanded to saying, okay, this is guessing that this one is DNA and guessing that this one is RNA. Now, if it's not clear, it might guess a different type than what you've intended. So say you intended for this one here to be RNA, then you'd have to specify that here. Now, hybrid strands are also supported in molecule conversion. So here we see this hybrid strand converted to molecule. And bonds can occur both within a given motif, within this DNA here, and across the motif strands of the sequence. So here we have the inner connection and kind of the outer one here. Now this form is also equivalent. So we can also describe that inner bond on the DNA as being, both corresponding to the first position. So the third term of the first position to the 6th term of the motif sequence in the first position. Now, this leads to sequence collections. A sequence collection is a set of sequences unconnected, except by, by further provided bonds or higher order features if they're connected at all. And this is expressed as a list in the first argument as seen here. So this is 2 sequences that aren't connected. As we see here. And here are a number of disconnected circular sequences. As before, this is a short form. And if you need to specify a type, you can add this BioSequence wrapper around any middle term here. Of course, disconnected sequences is an edge case, and we're more interested in the case in which they are connected. So here's an example with insulin, where we have, bonds, connecting these 2 insulin sequences together. And we can see that these bonds are disulfide bridges and obtain the corresponding insulin molecule. And both molecular and molar mass are available as properties for this sequence, even without converting to a molecule. And even more exotic combinations are certainly possible. Here's a collection with 2 bonded hybrid strands. You see them joined up here with their kind of 4 additional bonds. And we can generate molecules for these objects as well. If there are many bonds, spans can also be used in these more nested structures to conveniently specify, kind of a run of bonds. Here. Now, throughout we've been seeing example of BioSequence plot, but let's take a little look further. BioSequence plot has to handle kind of sequences of any size. So here we define a single pseudoknot, and here at this level, displaying each symbol makes sense. For larger sequences, these are better connected as tooltips, so that we can see, kind of where we are in this. And for very large structures, it's not practical to represent each term as a node, and so so many are grouped together here. So by default, every motif subsequence here is given a different color, and RNA and DNA are given different shapes. So here we see a sequence collection with 2 hybrid strands and a disconnected simple motif sequence. For more complex structures, the tooltips, which let's see here, become even kind of more complex to allow some navigation here. So we can see, sequence 1, motif 2 is DNA, position 2 is C. So now that, you've seen kind of the basic kind of representation, there are a lot of extensions, modifications, and other conveniences. So circular sequences are supported for all of these. We can enter them by standard abbreviation instead of by sequence symbol, which is sometimes easy to remember. We can convert circular sequences to linear sequences and vice versa. We can modify the sequence to add and remove bonds. We can add sequences to collections. And we can split disconnected collections so that, the first term and the third term are bonded together and the second one is not. So we can split that into, the 2 and 1 sequences that are actually connected. Now, sometimes it's handy to know if 2 sequences are identical, and this can be tricky, as you can represent the same sequence in different ways, or they can be in different order. So here we see we're taking a canonical representation, and that switches the order of these of these sequences here in the collection. Or say you have like, a hybrid strand that's actually just one motif, it'll simplify this to a single motif sequence. And it can also be handy to move bonds around. So here we move this bond inward to the innermost sequence. And this could be handy if you want to remove a sequence from there, but retain all of its local bonds. And similarly, you can move all the bonds to the outermost level, which is handy if you want to do some operation on all of the bonds at once. So in the future, we'll be interested in annotating sequences for coding regions and allowing those to be extracted with modifications or added directly with machine learning. Be interested in finding where a given sequence exists on other standard structures and finding the SNPs that might occur against those sequences and beyond. We're really fairly user driven. What folks need is what we're interested in. So as you're using this, if you find features you need, of course, please reach out. Good. So in terms of entities, we have of course gene, and then we're able to extract it in BioSequence. Likewise for proteins, you can obtain properties from it and extract those in sequences. SNPs are also provided. And we all of the built-in BioSequence types have have an entity so that you can see kind of what are the rules to find them and then see them in action. So here we can see the degenerate letter rules for B, and then when we ask for BioSequences of B, retain the same results. Genetic translation tables, same sort of thing. We can see the properties there. And then if you want to translate with respect to a particular translation table, you can use that to modify the translation operation. And recently we've been doing a lot with content updates to bring these to particular reference assemblies and expose that work better. And that's work we hope will be available soon. And we hope to do more to also expose the bond types that I've shown before so that those can be easily expressed and extended. So the Wolfram Function Repository has some great resources corresponding to all of these, as well as the data repository. So we could ask for a reference biosequence for the COVID genome and compare that against a sequence that we obtained from FASTA, and kind of align all that. So really the data and function repositories allow a nice extension to all of this. And there are some blog posts and further resources about that. So overall, we've been aiming to improve representation, algorithms, visualization, our knowledge integration through the entity system, our service connectivity so that you can enter things through fast IDs and have them brought in, what data formats are supported, what overall workflows you can do, and just continually express those communities and listen to see which of these areas are most important to develop. So really version 13 has been a big step forward for molecular sequences. Expressing all this higher order structure and visualization will really, you've seen enables us to express a lot more important biological systems. And we really thought that was the next logical step forward in order to express the structures that are actually out there in biological world. So I'm going to turn to feedback from questions. I'm going to look at the chat in Passable and see about any questions there. I want to thank all of the collaborators on this work. They've been tremendous. Okay, questions. BioSequence return as a graph, BioSequence graph function. Yeah, I think actually, or even like a graph property embedded in BioSequence itself. That's a great idea, and I think that'll come very naturally when as we expose more of the bond types. Then we can say, the individual sequence terms are connected by these primary bonds that are of this particular type, while these secondary bonds are of a different type. So as we expose more of the bond type system, we'll be able to publish graphs that are annotated in a sensible way. So that's kind of the dependency order there, but that makes a lot of sense. So that's a question about would there be a benefit to having a BioSequence graph function returning a graph? And I think that's a great BioSequence property in and of itself, and something we've definitely thought about as we've introduced these bond types and bond representations. Nice to have sequence structures. Oh yeah, absolutely. So that's when I talk about annotations, not only for coding sequences, but for labeling, sequence structures. That's a huge application for that, and particularly, so that annotation feature kind of discussed in the future talk, that fits very well. So for alpha helix and beta sheets, yeah, that fits in very well. And of course, having, that be conversant with the bond system is something we think a lot about. Let's see, your project. Oh yeah, that's great. How complicated of conformations or cross-linking this might be able to handle. It's in, I think representationally, we've tried to make it pretty flexible, so that for, by having untyped bonds between singular positions, you can, we really tried not to necessarily impose it was really a tightrope between imposing too much semantics and not being enough that we could generate, we couldn't generate molecules from it. So we allowed a lot of flexibility in terms of, what the users could specify. And then for operations that really required it to be rigorous, then we would kind of lock things down. But I think we could go further if we had to. Like, you could imagine like a bond with 3 or more terms, if the, things are really connected in kind of like a complicated way, that you really have to have all kind of the terms involved. I think things like that are not off the table. Now, on the other side, so I guess the short answer is it's pretty flexible. It's not as flexible as it could be, depending upon, what applications are really in demand. So try it out, and if you find you're stressing it, certainly let us know. I'm excited that you said that you're going to be using it for a project. That's wonderful. And also, this will be some stress tests for other operations. Like, okay, great, you specified it. What does BioSequence plot do to it? That's something we're also kind of looking to refine. It's not only representation, but that the representation is handled nicely by all the supporting features. So yeah, definitely stay in touch, and we hope that you can use that going forward. Great, let's hear. I think I've addressed all 3 of the comments here, and I've just about run out entirely of time. So thanks everybody. This has been great. I'll be on the Rocket chat, and I'll keep watching this chat here. So if anything comes up further, feel free to type in there, and hopefully I'll be able to catch it before too long.
Yeah, thanks, John. If anybody does think of any questions, like you said, you can type them back in that chat. That'll be around for 30 days after the conference is done. So, even in 2 weeks, you can come back and put something in there, and we can get you an answer. So if there's not anything else, I appreciate everything, and everybody have a good rest of the conference.
Thanks, Brandon. Thanks, everybody.